and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with virologists, students, and postdocs that belong to the American Society for Virology so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On October 5th, 2021, we talked with Dr. Lisa Bauer, a postdoc in the Van Riel lab at the Erasmus Medical Center in Rotterdam. Lisa obtained her master's in microbiology and molecular immunology and her PhD identifying and characterizing therapeutics for enteroviruses. She is currently using airway organoids and neurons differentiated from pluripotent stem cells to investigate how infection with respiratory viruses, such as Influenza A virus and SARS-CoV-2, affects the central nervous system, both acutely and long-term due to post-infectious sequelae. So nice to talk with you today. Why don't you tell us a little bit about yourself?
Hi, so first I want to thank you to give me the opportunity to talk with you. And I think it's a very nice opportunity for young scientists on this platform to also see behind the curtain who we are and what we're doing. So my name is Lisa Bauer, and I'm a postdoc in a group of Debbie van Riel at the Erasmus Medical Center currently. And I come originally from Austria, from a very small little town where we are normally around, under the week, let's say, 500 inhabitants, and on the weekend we're around 1,000, so we are kind of a hotspot for the Viennese people to come and have a little bit a quieter time and escape a little bit the big city. And I was very fortunate that I could kind of escape, let's say, my little hometown and go to the big city and study there and get my education and also go to school there. So that was very adventurous already.
And how did you first become interested in science? Like, sort of what got you interested in it?
So in science, I always wanted to understand when I saw, so my mom has on her left arm, she has a little scar, which back then I didn't know what it is. And I always asked my mom, why do you have that? And my dad, but I don't have that. And my mom was saying, oh yeah, that's about against pox viruses. I'm like, what is that? I don't understand. Can you explain me? But it took a very, very long time until people actually really could explain me what a virus is, like this whole big concept. And I think then my curiosity from there just grew more and more. And also already in primary school, I was more interested in learning about, as we call it in also like children's diseases, than, for example, mathematics or German or something like this. So I always wanted to know a lot about that. And yeah, and already from very on, I just wanted to know why we can get vaccines, what are they helping against? And yeah, so, and that never stopped until today.
Okay, and then can you tell us a little bit about sort of the path that you took in your education? So, how you got to where you were in sort of undergrad and then into your PhD and now into your postdoc.
So I did, as I mentioned before, my bachelor and my master's studies at the University of Vienna. And before this, in 2006, I finished my A-levels. And the main topic of my A-levels actually doesn't matter if it was biology, English, or whatever. It was about pandemics, because back then, so it was in 2006, there was the wave of SARS-1 and also bird flu, how we call it in German, so avian influenza. And I just took the opportunity to expand there a little bit my knowledge. And then I was talking with people in my school and teachers with what is the right path for me, because I really wanted to study viruses. And then one of the teachers came to me and said, oh yeah, then you have to study pharmacy. So I started to study pharmacy at the University of Vienna. But after a year, I figured out that that's not what I'm interested in. And then I again talked with other people and they said, yeah, why don't you try chemistry? So I studied a year of chemistry. I really liked it. But again, there were no viruses until I figured out that we have a side branch, which was microbiology, which was embedded in the whole biology. And that's then I followed during my bachelor's. And there we had, I still can remember, it was the microbiology course #2. And then we had this professor coming in, Timothy Skern. And he was like, okay, what kind of diseases do you people know? And we were like, yeah, polio, pox viruses. And he was the first one who really brought to me the concept of viruses. And then I knew, yeah, that's exactly what I wanted to do. And so I did then also my bachelor internship in his lab. And that was so he's actually a structural biologist focusing on viral proteins, especially from the family of Picornaviridae. And from there on, I then went on to do my master's, which was molecular microbiology and immune biology. And I did my master's internship at the Center of Molecular Medicine in, let's say, the only lab I could find in Austria, in Vienna, which was working on virus-host interaction. And there I worked mainly on more in the interfield between innate immunity, epigenetics, and virology. And then I was looking for PhD positions, and it was not the easiest tasks, let's put it like this. And so for 1 year, then while looking for PhD positions, I was working in a plant molecular pathology lab. So I worked with bacteria and with fungi infecting maize plants. And it was also very fascinating. And then I got their PhD position offered and I declined because it was not about viruses. I told him, so if he could find a virus for maize plants, I would be happy to stay. And then luckily I found a PhD position in a Marie Curie network. So it's funded from the European Union. It's a very big network. So we were in total 15 PhD students from different kind of fields. So from virology, medicinal chemistry, and more from the technical background even. And our main goal was to produce antivirals against mostly plus-strand RNA viruses, but also we had one project running on RSV. Yeah, and then I finished my PhD, and I went very smoothly on to my postdoc, also in the Netherlands. So I did my PhD in Utrecht, and then I moved from Utrecht to the Erasmus Medical Center, and I work now on influenza and enterovirus T68, and we want to kind of to understand how respiratory viruses can invade the central nervous system, and of course nowadays also with SARS-CoV-2.
Okay, wow. And can you tell us a little bit about, so I guess in your postdoc, what were you looking for when you were looking for a postdoc lab? So sort of what defines a good postdoc lab?
There are two things. I heard from a lot of people that if you want to really succeed in science, you should go to a very big lab, a very well-known lab. And I think I did exactly the opposite. So I came from a very well-established picornavirology lab. And Frank van Koepenveld, I think everyone who is working with picornaviruses, everyone knows him. But for me, I wanted a bit more, I wanted to completely switch gears to learn first, to broaden my horizon. But I also wanted to help a young lab to set up a new expertise with maybe an expertise I have. So this was one reason. And the other reason, so far, I also had during my whole career, supervisors were only males. And I think as a female scientist, you also should see on how the world looks from a female perspective, which is, I think, for males, not always completely clear. And that was my second point, especially also the connection during the interview what I had with my boss now, with Debbie. I think that made it for me clear, besides, of course, the research project, that this is a very welcoming and friendly environment where we work hard, but we also party hard, kind of thing. So that was for me very important.
Great, great. So why don't you tell us then a little bit about the work that you're doing now, maybe talking a little bit about some of the techniques that you're using, some of the big questions that you're trying to answer, things like that.
So I came to the lab. So Debbie is very well known in the field of Influenza A viruses, which is completely different to my little small, not enveloped picornaviruses. And she needed someone who to help her set up a lot of molecular virology techniques, let's put it like this, for enteroviruses. So titrations, how we can we grow the virus, can we make infectious clones and stuff like that. And so I helped a lot with that. But my main focus now is on influenza, and especially we want to understand how these viruses, in general respiratory viruses, how they can go systemic. So why do we find, for example, also now with SARS-CoV-2, why do we find them in the liver, in the heart, in the brain? So we are specialized, especially in the brain. And so we use a couple of different techniques. So one technique is what we have is that we use airways organoids, which we then culture from 3D, we culture them then on 2D, so that you have a very nice pseudostratified layer of epithelial cells. And to understand, so if you think, especially for flu, if you have a pneumonia, you need to go to the alveolus, right? So you have a very tight connection from the epithelial cells to the endothelial cells. So I was very busy now setting up a model on the transwell with these organoids, where you have on the apical side the epithelial or the organoid derived epithelial cells, and on the basolateral side lung macrovascular endothelial cells. And we want to understand how this interaction between epithelial cells and endothelial cells actually can form, for example, cytokine storms and stuff like this, and what actually triggers the endothelial cells. So this is one part of my research line. And the other part of my research line is that we're using a lot IPS differentiation. So we have induced pluripotent stem cells, and we can differentiate them in different neuron lineages, let's put it like this. So we have now established in the lab very nicely, we can make cortical neurons, and where we supplement these cortical neurons with our own differentiated astrocytes. And there, in combination in these co-cultures, the neurons are actually very nicely active and can fire in their synapses. And we want to understand a little bit more the electrophysiology of these neurons, especially what happens then with virus infections, and then maybe even compare. So now we are focusing more on SARS-CoV-2 and flu, but also maybe later on compare this with real neurotropic viruses, like for example Zika or West Nile. I think that would be very nice. And since we're also working on enterovirus T68, we now established differentiations of motor neurons, which works very nicely. And then we want to study a little bit the pathogenesis of enterovirus T68 in these motor neurons a bit more in detail, and also looking more behind the electrophysiology. So I think overall, with all of this, the aim is, so what I'm really interested in is to understand how, so let's start from SARS and flu. So we know for both viruses that it can cause during the acute time of infection quite severe, sometimes neurological complications, but also after the acute phase and the post-acute phase and post-infection, a lot of neurological sequelae happen. And I think it's absolutely not understood how this works. And I think that's very, very important to understand how this works. So we're now driving a bit more into this direction and to more understand on a broader perspective how this actually happens.
Right. And can you talk a little bit about, so there are viruses that are sort of like bona fide, viruses that are known to invade the CNS, things like West Nile, even though obviously it's a rare sequelae in a way, but still they are done, bona fide neurotropic viruses in the CNS itself. And then, as you were alluding to earlier, there's viruses like flu or potentially SARS, where it's still not clear that they're really infecting the brain per se, or at least neurons in the brain, but they're having these effects in the brain. So can you kind of talk a little bit about what might be the differences between the viruses that would actually set them up like that?
So for example, with these real neurotropic viruses, I think what you see already just in vitro data is that these viruses replicate very efficiently. So they can replicate in astrocytes, in neurons. I'm not sure that I think so far there's not too much data out there about microglia because they're quite challenging to get into culture. But these viruses replicate very efficiently and they can spread throughout the brain. And on the other hand, you have SARS-CoV-2 and also influenza A viruses, where we see in our in vitro cultures that these viruses can infect very good neurons. So, I mean, SARS, not that good. So if, for example, if we infect our cultures with SARS, we every now and then, very scarcely, we will see one infected cell. If you compare this, for example, to influenza A viruses, especially H1N1, we see if we infect these cultures, we see almost 90% of the neurons infected. But both viruses, they cannot replicate efficiently, so they never produce any progeny viruses. But still, what we see is that at least with SARS-CoV-2, we showed recently in a paper that even though you have very rare infection in these co-cultures, it still triggers a mild immune response, which may be, well, it's only a little piece of the whole answer, but maybe that also explains why we see this acute and post-acute neurological sequelae, that you just have an induction of neuroinflammation by the presence of the virus, which not necessarily needs to be efficient replication in the case of SARS-CoV-2. Because, for example, we also have the influenza A virus, the highly pathogenic H5N1, and we see that this virus replicates to very high titers in our in vitro cultures, and also in ferret brains, for example, it just spreads throughout the brain and induces massive amounts of inflammation. So, but to really understand this, we're, I think, in the very first beginning on what is really going on there. Inefficient replication, or at least the presence of the virus in the neurons, for sure leads to something.
Right.
But yeah, there's a lot of, we can speculate a lot what will happen. So I think one contributor to all of this are cranial nerves, which I think people underestimate always a little bit. Because if we think of, for example, these viruses replicate very well in the nasal epithelium and the olfactory epithelium, right? And this epithelium in our nose is just innervated by a lot of different nerves. For example, we have the olfactory nerve, right? And we have the trigeminal nerve. And for example, for flu, we see infection of the trigeminal nerve in animal models, as well as the olfactory nerve. So I think, especially with the olfactory nerve, that also has been shown for SARS-CoV-2 as a potential entry route for these viruses, which I think is often a little bit neglected, because people always think, oh yeah, you need to get either through viremia. And I mean, also with SARS-CoV-2 and also with flu, even though it's a little bit underrepresented, I think there's actually quite some substantial viremia, which also contributes. So there's a lot of factors, but definitely cranial nerve play a very important role to distribute these viruses to the CNS.
Right. Great. And then I guess for when you're using all these organoid cultures, so how do you look at viral infection, and how do you look at the changes that are occurring? Are you using a lot of sort of like I don't know, epifluorescence, like sort of image-based analysis, or are you doing more things like single cell or RNA-seq or qRT-PCR? How are you actually looking at the virus and the host?
Yeah, for viruses, so we mostly use titrations, of course, or for SARS-CoV-2 also plaque assays, which I like a lot. I think it's a very nice technique, a plaque assay. It's fantastic. And to evaluate more the host side, let's put it like this, we use a lot of imaging. And yeah, as you said, also not yet single cell RNA sequencing, because it's very expensive, and yeah, you always need to find a little bit of funding for that. But bulk RNA sequencing we do a lot, and qPCRs. And we also have especially for for cytokine or interferon expressions. They have very nice tools, which is called LMtV. It's a multiplex bead assay where you can measure up to, I think, 13 or 15 cytokines at once. It's a fax-based assay, and that works actually very good in our hands.
Right. Cool.
And then you already get a very profound profile of what is happening during the infection.
Right. So what do you think about sort of for your future? Where do you see yourself going in the next couple of years?
I would love to go back to Austria, to Vienna, because I think also during this pandemic that there's almost no virology research, or at least it's not very present. And I think that is really a shame. And I would like to contribute there also in my home a little bit to a more understanding on virus diseases. So ideally, I would like to stay in science, but yeah, we all know that this probably is a dream. But if you do not try it, you will never know it. But in the end, I'm open to a lot of things. I would wish for myself that I could work until retirement with viruses because they are really fascinating.
Yeah. And so in Austria, what is the sort of the system to get funding and things like that? So if you want to be a scientist or have a lab in Austria, what is the system that people follow?
Yeah, so we have a couple of research institutes for fundamental science, and they have their funding from the Austrian Academy of Sciences, and they are funded by taxpayers' money. Let's put it like this. And we also have the FWF, which is, I think, very similar to the NIH, where you can just apply for funding. So you have a certain amount of money where you can apply for funding for all groups of sciences. And yeah, that's basically it. So the science funding is not too big. And of course, you also have the chances through the European Council to get some money through the European Union. But yeah, it's really tough. And I'm not sure how the science money in Austria will develop, or how our government will fund sciences, especially now after all of the debts what we have now since the corona pandemic. Just hope that in general, politicians do not forget that you need fundamental science in order to have a well-running society, because I think this pandemic really showed how important science, and especially fundamental science and academia, is to fight this pandemic. Especially in Austria, most of all of the PCR tests in the beginning, the sequencing, everything was run by academic labs. And I think that should be a bit more appreciated.
Yeah. And how is Austria doing now with the pandemic?
I think overall they handled it relatively good. One of the problems what we have, and I think it's not true for all the European countries, but for us, we have a very high rate of people who do not want to get vaccinated, either out of fear or they're just anti-vaxxers. And I think our vaccination rate currently is at 64%, and it's very complicated to push up these numbers. And I think also no one really has an idea. So I help a lot with educating my friends if they have problems or if they have concerns regarding the vaccine. But yeah, there are also, my hands are a little bound, and convincing an anti-vaxxer, I think, to get vaccinated is pretty pointless. They are just too indoctrinated with not getting the vaccine or any vaccine at all. And you have to respect it too.
Yeah, I think we have the same challenge here. There's different groups in a way of people. And so I think you have to really tailor your messaging and your outreach and your potential, solutions or what you're offering to, very specifically in a way. And so that's always challenging. We always like to sort of have a one case fits all kind of strategy, but that's clearly not working, I think. And so, we have similar problems. We're, at some point, we'll have to figure, there's a science in this too. And so I do believe that there are solutions. We just have to figure out what they are.
Yeah, I have to say, I have to say in Austria, they're also quite adventurous. So for example, they provided a lot of vaccinations in churches. So you just can go on Sunday to the mass. And then after the mass, there's just a bus standing in front of the church and is vaccinating people. And I think that worked actually quite good, especially in the rural regions. And you also have this big discrepancy between, I would say, the big city and the rural regions. And it's not only because it's harder to reach them with the vaccines, but I think it's also maybe an education level, because you just don't reach them that easily.
Yeah. And you sometimes wonder if there's also a general distrust of the central government. We have that here in America as well too. So it's sort of like, how do you reach people in a way that is non-governmental, non-political, and are just sort of directly able to speak to their needs as people? It's difficult.
It's very difficult and also very emotional. And that makes it very hard to also discuss with people, because I don't want to offend something. And I think as a scientist, we are very happy to have heavy discussions, and we are okay with agree to disagree, right?
Right.
And sometimes the discussions what a scientist has is not always relatable for other people, because they want to bring you on their side of opinion.
Right.
It's not so easy for them to agree to disagree.
Yeah, I agree. So what has this past year and a half been like for you? I guess talking a little bit about the pandemic, did you have to go through sort of a shutdown in your lab, or what happened?
Yeah, so I started in the lab of Debbie on the 1st of March in 2020. And I went to a defense in Belgium. That was in the second week of March. And on the day when I returned, I entered the lab and everyone was sitting in a circle. And we were saying, we're in a lockdown now. And we were all so sure that probably in a month or two, everything will be better. Little did we know. So, but for me, it was relatively soon we started up with all of the diagnostic work. But back then I was living still in Utrecht, and that's around, I think, 60 kilometers to Rotterdam. But it's very easy connected with the train. So it took me like 1 hour, 10 minutes. And in the beginning they said, I still can come and help with the diagnostics. And then they said, maybe it's better if I don't commute. And in the middle of the lockdown, I also then moved to Rotterdam. And then it was easier for me to again go to the lab and help stuff. And I think we were in lockdown until the mid-May or something like this. So May, June. And then slowly but surely we were coming back. And in the beginning, it was only SARS-CoV-2 related research. And then at a later point, around August, we also could start up other research again. And then during even though we had a second lockdown in the country, but our lab actually never went into a second lockdown. We were just making sure that if you have to do lab work, you can go to lab, but work from home as much as possible. Right.
Yeah, we've done something similar, I think, from the get-go. Yeah.
And it is very weird. I mean, I started now 1.5 years ago, and I still don't know everyone, because not everyone, especially the bioinformatics people, they're still not coming every day to the lab. So, and the virus science department at the Erasmus Medical Center, we're a lot of people. So I still don't know everyone, and that is a little bit awkward. Let's put it like this.
Yeah. We have the same thing. So, people that you should know, but you don't actually in person.
Yeah.
Yeah. All right. Well, thanks so much for talking with us today. We appreciate hearing about your research, and good luck on your organoid systems. And we hope to hear some interesting stories from you.
Yeah, I hope so too. Thank you very much. It was very lovely talking to you.
Podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcast providers, or at lmtv.podbean.com.